On behalf of everyone at 908 Devices, I would like to welcome you to today's webinar presentation. The topic of today's webinar is Simplifying Oligonucleotide Characterization with Microchip CEMS. I will first provide a quick introduction to 908 Devices and what we do. I'll then give an overview of the Microfluidic CEMS technology, also known as the ZipChip. Then we'll look at the current methods used for characterization of oligonucleotides and the challenges associated with these methods. And then I'll present oligonucleotides data that was acquired on the ZipChip. So let's get started. So the company has three major product lines: the MX908 shown on the left, the Rebel in the middle, and the ZipChip on the right. The MX908 is a handheld mass spec for field-based on-scene analysis for the detection of a variety of compounds such as chemicals, drugs, priority toxics, and explosives. The Rebel is a desktop device that has a microfluidic capillary electrophoresis and mass spec built into a single device. It is a purpose-built device used for accelerating bioprocess development for a variety of applications such as protein therapeutics, cell and gene therapies, synthetic biology, cell culture QC, and bioprocess analytics. But the focus of today's talk is the third product, which we call the ZipChip. So let's zoom in on ZipChip. What is ZipChip? In a nutshell, ZipChip is a front-end separation device for mass spectrometry. It is a microfluidic device that integrates capillary electrophoresis with electrospray ionization, all on the same device. The ZipChip interface shown here weighs about 8 pounds, and once mounted on the mass spec, can automatically sync and align with the mass spec inlet. The separations on a ZipChip occur freely in solution, and samples are quickly separated within minutes to provide sharp, well-defined peaks. And these peaks are then detected by mass spec analysis. The ZipChip interface is currently compatible with mass spec vendors such as Thermo Fisher, SciEx, and Bruker. The cassette shown in the center of the screen is the actual microfluidic device, also known as the ZipChip, which is actually at the heart of the ZipChip technology. This chip integrates capillary electrophoresis and nanoelectrospray ionization into a single microfluidic device. The chips are available in two different formats: high-resolution chips for applications that require greater separation between analytes, or high-speed for high-throughput applications where speed is more important than the separation resolution. Each chip can perform up to 125 or 250 injections. We also have the reagent kits that cover a broad range of applications, from small molecules, peptides, native proteins, and most recently, oligonucleotides. These kits contain background electrolyte solutions and diluents that are optimized for specific applications. The kits have been tried and tested in our 908 labs and produce efficient separations and stable electrospray. The main purpose of having these kits is to take the guesswork out of your analysis and eliminate the need for method development and to provide a ready-to-use solution for your analysis. Let's take a closer look at the microfluidic chip itself. So as shown in the schematic here, the chip has 4 different wells. The sample well that holds the sample. There are 2 BGE wells, or background electrolyte wells, that hold the background electrolyte solution and are connected to electrodes for capillary electrophoresis. And the 4th well is the waste well. There's also the serpentine channel in the middle that performs electrophoretic separations. The chip has three main functions. Number one is sample injection. Number two is electrophoretic separations. And number three is nano electrospray ionization. The ZipChip uses a novel sample injection method that utilizes pressure-driven flow to precisely deliver small aliquots of sample into the separation channel. Once the sample is injected into the injection cross shown here, high voltage power supplies are turned on and analytes migrate through this serpentine channel, the separation channel, and separate from one another based on differences in their electrophoretic mobilities. Once the analytes migrate up to the sprayer corner, they get electrosprayed directly into the mass spectrometer and are detected. And because the sprayer potential is held roughly at plus 3.5 kilovolts, all analytes, including even the oligonucleotides, are detected as positive ions. So what about applications? ZipChip is a truly versatile platform for analysis of a wide range of molecules. The application space ranges from small molecules to peptides to intact proteins and beyond. The platform has been widely adopted in biopharmaceutical industry and leading research labs for a variety of applications. And just in this year alone, in 2021, there have been several peer-reviewed publications highlighting the unique aspects of ZipChip. And some of these publications have been highlighted here. We have recently expanded the applications toolkit by adding oligonucleotide capabilities, which I will go over in greater detail in the next few slides. So oligonucleotides, or oligos for short, represent a distinct class of therapeutics that include molecules such as RNAs, DNAs, and their structural analogues, and are effective against a wide range of disease conditions. So synthetic oligos have gained significant interest from the biopharmaceutical industry in the recent days and years, and several of these candidates have been either approved or have progressed to later stages of clinical trials, including some of the leading vaccines for COVID-19. Oligos are single or double-stranded linear molecules and are present as different modalities, such as antisense oligonucleotides, siRNAs, miRNAs, and aptamer-based therapeutics. So these are polyanionic molecules that exhibit very different chemical and physiological properties compared to the traditional small molecules or protein-based drugs, and pose a different set of challenges for analytical characterization. There are currently 14 different approved oligotherapies on the market, and a vast majority of them are based on antisense oligonucleotides. And just to highlight the recent spike in interest in this field, there are 61 different oligo-based therapies in clinical trials just for oncology alone. So what are some of the common analytical techniques used for oligo characterization? Among all the mass spec methods used for oligos, not surprisingly, LC-MS remains the most dominant and widely adopted method. LC-MS can be used for several different oligo analysis workflows, such as identification with molecular weight, sequence length confirmation, impurity analysis, and even metabolite identification. And among the LC methods that are used, the most widely adopted separation technique is reverse phase LC with ion pairing agents. Even though this method is capable of producing good separations and peak shapes, there's a major drawback associated with this method. Ion pairing agents tend to contaminate LC fluidics columns as well as the mass spec system. And as a result of that, extended cleaning procedures are required before switching to other applications to avoid cross-contamination of ion pairing agents. In a lot of cases, an entire system is sometimes dedicated to just the oligo workflow alone to avoid switching between methods, which may not always be the most economic solution. The other separation technique that is gaining popularity these days is HILIC. HILIC methods don't use ion pairing agents, but the method itself can be finicky, and it often requires long column equilibration times to get reproducible results. And this is where ZipChip shows a lot of promise, as it can overcome several of these disadvantages. So what are the key benefits of ZipChip for oligo analysis? First and foremost, the ZipChip background electrolyte solutions do not use any ion pairing agents, which makes switching between methods very easy, and it also eliminates long downtimes associated with instrument cleaning. So you could be analyzing oligos in the morning, for example, and native proteins after lunch without any problem. ZipChip can be easily coupled with high-resolution accurate mass instruments and can thus provide identification and quantitation with accurate mass. And as described before, ZipChip separation mechanism is based on electrophoretic mobility of the molecules rather than hydrophobicity. And that is why the ZipChip is an orthogonal technique to the traditional LC approaches. ZipChip methods are fast due to the microfluidic technology, and you can expect to generate data within just a few minutes. And I will show you some examples in the next few slides. And finally, there's no method optimization required, and the same method is applicable for a wide range of oligonucleotides. All right, so the ZipChip MS method for oligos uses a chip with bare glass that is silica in the separation channels, and a background electrolyte solution from our Oligos kit, which has a pH of around 8.5. So we chose the high pH for this application because the high pH of the solution ensures a steady electroosmotic flow in the separation channels. You can use both high-speed and high-resolution chips for this application, of course, both with bare glass chip chemistry. But for all the work that has been presented here, a high-resolution bare glass chip was used. Now, the silica surface in the channels is negatively charged at pH of the background electrolyte. So when a voltage potential is applied, or a potential difference is applied across the separation channel, it induces a steady unidirectional flow of background electrolyte, resulting in electroosmotic flow. So this strong electroosmotic flow results in the migration of all analytes towards the ESI sprayer on the chip. And positively charged species in the samples migrate first, followed by neutrals, and the neutrals are then followed by negatively charged species, which are trying to flow in the opposite direction, but they get pushed down the separation channel because of the EOF. So for the initial experiment, we chose a very simple analyte, a 10-mer single-stranded RNA molecule that's shown here. As shown in the electropherogram, the HRB chip, or the high-resolution bare glass chip, enabled easy analysis of negatively charged 10-mer oligo within just 5 minutes. And we were able to get sharp peak for this analyte that was just a few seconds wide, and also showed a Gaussian distribution. The mass spectrum for the 10-mer is also shown on the right. Now, if you look at the mass spectrum, the biggest difference in the mass spectrum compared to negative mode analysis is that there are fewer charge states observed in the positive mode spectrum. And this potentially results in enhancement of the sensitivity of this method. And of course, all of this was accomplished without the use of any ion pairing agents. So after getting encouraging results for a 10-mer, we decided to use the same method for analysis of slightly larger oligos. So we looked at two different molecules, a 20-mer single-stranded RNA and a 4-dimer single-stranded RNA. And both of these molecules were unmodified. The figure on top shows the data for a 20-mer, and the figure on the bottom shows the data for the 4-dimer. And once again, we observed sharp peaks for just a 1 nanoliter injection for these samples and generated clean mass spectra. The insert in the mass spectra show a zoomed in view for a +4 charge state for the 20-mer and a +5 charge state for the 40-mer RNA. The MS method parameters, such as the orbit trap resolving power and in-source CID, should sometimes be optimized to obtain optimal peak intensities for depending on the size of the oligos. But the ZipChip separation method essentially remains the same. There are no changes required to this method. In all of these samples, we observed that the protonated adduct was the most prominent adduct. That's shown in the zoomed in view here. And we also observed other alkali metal adducts such as sodium and potassium. But in general, the observation was that the level of adduction went up as the size of oligos also went up. So what about separation? So the separation efficiency of this method was also assessed. And to do this, we ordered several 20-mer single-stranded RNAs from IDT, a commercial vendor. And the modified standards based on that same nucleotide sequence were also obtained. And these modifications included phosphorylation, biotinylation, and glycan spacer, consisting of a di-deoxyribose spacer. So we prepared a mixture containing 500 nanomolar of each of these modified oligos. And as you can see from the electrophoregram, the phosphorylation and biotin modified RNAs were baseline resolved from the main or the unmodified peak. And the glycan modified RNA appeared as a shoulder to the unmodified species, as shown in the electrophoregram. So it's a really nice method for separating structurally different oligonucleotides. So in a separate experiment, 100 nanomolar phosphorylated 20-mer RNA was spiked into a 1 micromolar solution of the corresponding non-phosphorylated 20-mer. And due to the presence of an additional negative charge from the phosphate group on the phosphorylated molecule, it was detected as a baseline resolved peak from the non-phosphorylated molecule, as shown in the electrophoregram here. And the resulting mass spectra for the two peaks are also shown. The order of migration is in accordance with the net charge on the molecules. So the more negative molecule migrated later, which is the phosphorylated version of the molecule. And we were also able to generate clean mass spectra for both. So after demonstrating the separation capabilities of this method, we focused our attention on determining the robustness and reliability of this method. To do that, a 1 micromolar solution of 20-mer, the same 20-mer single-stranded RNA was repeatedly analyzed for a total of 375 injections over 3 consecutive days. And for this work, 3 different HRB chips were used with 125 injections per chip. A representative plot for one set of experiments of 125 injections is shown here. The data clearly demonstrates the high migration time and peak area reproducibility of the ZipChip oligos method. And as shown here, the relative standard deviation for peak area was 14.9%, and the RSD for the migration time was 1.4%, and which is pretty good. These numbers are consistent with other standard applications on the ZipChip as well. The sensitivity of the method was tested using the same unmodified single-stranded RNA standard. And the standard was serially diluted from a stock solution to generate different standards ranging from 5 nanomolar to 10,000 nanomolar. And these dilutions were made in the background electrolyte itself. Each calibration standard was injected in duplicate. The calibration curve that was generated is shown here in this figure. And the method was found to be highly linear within the dynamic range of 5 nanomolar to 10,000 nanomolar. And the R-square value was pretty good, as seen here. The limits of detection and quantitation were also determined. And based on this data, the LOD for the 20 mer was 5 nanomolar, and the LOQ was 10 nanomolar. Now this shows that the method is highly sensitive, and there's good linearity. The good linearity of this method should be able to give the users an idea of the relative quantities of impurities that are present in the sample over the same dynamic range. So to determine whether this method can be extended to analysis of oligos with higher order structures, a single-stranded RNA with 21 bases, along with the same 21 mer RNA, which was annealed as a duplex with its complementary antisense strand, so a 42 mer, was also analyzed. And these samples were either diluted in the BGE or DMSO. When the duplex was diluted in the BGE solution, we obtained a large peak for the intact duplex, as shown by the green trace on top. And this clearly shows that the gentle conditions of the BGE do not disrupt the secondary structure of the duplex. Lesser amounts of the sense and antisense strands were also observed in the sample, and that suggested that these strands were probably present in the sample itself. In the next experiment, the same sample was diluted in DMSO this time to chemically denature the duplex, and then it was loaded on the ZipChip. And this time, strong peaks for sense and antisense strand were observed, with very little duplex. And this data clearly indicates that the duplexes can be very easily analyzed using the ZipChip platform without significant changes to the separation method. So to summarize today's talk, ZipChip is a microfluidic device that seamlessly integrates capillary electrophoresis with mass spectrometry. And it offers unique advantages for a wide range of applications, from biopharma to omics to cell and gene therapy, as seen from this oligonucleotide analysis method. The ZipChip Oligos kit offers a hassle-free method for oligo characterization using high-resolution accurate mass with good sensitivity and linearity over a wide dynamic range. The kit also eliminates the use of ion pairing agents, which is a big drawback for LC methods. The kit provides an easy way to perform analysis for intact mass, for impurity analysis, and for oligos with higher order structures. And finally, it requires minimal method development. With that, I would like to thank all of you for your attention.
Great. Thank you very much, Adi, for that great presentation. Okay, so moving on, we now have time for a Q&A session where Adi and Scott will take some questions from the audience. Don't forget, you can still submit your questions using the questions panel that you'll find in the menu on the right side of your screens. So let me take a look at the first question. We have one here. Somebody is asking, can impurities be separated from the AP main peak? Who can answer that one?
So I can take that one. Impurities can be analyzed using a CE-based separation as long as there is as long as the impurity can be electrophoretically separated from the main species. So if the mobility of that species is different from the API, for example, I showed one example of the phosphorylated impurity that clearly has a different charge state compared to the API, and that can be effectively separated on the ZipChip.
Excellent, thank you. Okay, another question. Somebody asks, does the peak width allow MS analysis? And if so, how many MS runs for each run? What would you say?
I could probably
So
take
the
that
short
one.
answer is, yeah, go ahead, Scott.
Yeah. So yes, the peak widths that we generate are, 3 to 4 seconds wide for this analysis. So they're, it's fast, but it's not so fast that MS can't be done. Obviously, it depends on the specifics of the instrument, but there's nothing about this that is too fast for that.
Okay, great. And Adi, anything to add on that?
Yeah, I'll just add that you can also modulate the peak width by reducing a parameter called field strength, and that will make the peaks a little fatter without losing the separation efficiency, and that can improve the duty cycle for MS analysis.
Okay, good. Okay, we have somebody asking a question regarding the RSD percent. For RMT, the RSD percent was 1.4. However, what about the RSD percent for the uncorrected area for the peak of interest obtained? Can you provide more information?
Sure. The data provided was actually for it was uncorrected. It wasn't relative migration time. It was uncorrected migration time. So the 1.4% number is uncorrected migration time RST, not relative.
And also, Adi, the peak area data that you showed was also just raw peak areas that were measured. There was no correction there.
That is correct.
Great, thank you both. Moving on to another question. Somebody is asking, do you have any examples for ladder sequencing?
Yeah, I can handle that one. So the CE separation is a totally different mechanism from the LC separations that you might be used to. So the LC separations are generally length dependent. So, if you increase the length, you increase the migration time. That's not how this works. So the separation is mobility based, which is a function of charge, and size is involved. But as oligos get larger, they also, every increase in length adds more charge. So there actually is no direct correlation. That being said, there is still differences in mobility that are based on the chemistry of the molecules and the specifics of the structure. So it obviously separates things, but not specifically on length. So if we do a ladder, we actually tend to see some separation, but it's not in order of the length.
Okay, good, thank you. So moving on to another question. Somebody asks, can you confirm if this was performed in positive mode LC-MS? Who can take that one?
So we haven't done that work ourselves, but our collaborators in the industry have, and they reported that they saw comparable results in positive and negative mode in terms of the sensitivity of the method and also the level of adduction.
Excellent, thank you. Okay, so another question. Somebody is asking, Did you ever analyze crude material for failure sequence? Or how good is your chromatographic separation power for separating full-length API from N1 and N2? Who can take that question?
I can do that. I mean, so that the answer to that is a bit similar to my last answer about the length not being the direct driver of the separation. So in some cases, we will see separation between the main peak and an N-1, N-2. That's obviously easier the shorter the primary species is as it gets longer, then the differences associated with any change is smaller. We do, of course, have the mass spectrometry data, so even if we don't have full baseline separation, there is information from the mass spectrum, but not in all cases, right? So if your primary application is I need to separate the N-1, we certainly wouldn't promise that this will do that in all cases.
Good, thank you. Okay, another question here. Somebody says that they have a ZipChip device and was wondering if they could use this kit for their analysis. So for example, E. coli total tRNA purified treated with RNAse T1 would generate smaller fragments in the range of T to 10 NT. So can these be analysed by use of your kit and the ZipChip? Does that make sense to you to give an answer?
It does to me, yeah. And this is still a new application for us, so we can't say for sure about the details of it, but yes, on a basic level, that application should be a nice fit for this. Since they already have a ZipChip, certainly they should talk with their field application scientist, and we can work together to get that going.
Perfect, thank you. Okay, moving on, another question. Somebody asks, could ETD be a reasonable technique for oligo sequencing despite the relatively low charge states? Who can answer that one?
Yeah, I'll take a crack at it. We don't really know for sure yet. So we haven't run this on any instruments that have ETD fragmentation capability. I think there's no fundamental reason why it can't work. As Adi showed, we are still generating, multiply positively charged ions, and so we don't know for sure what the efficiency of the fragmentation will be, but it certainly could work.
Brilliant, thank you. Okay, another question. Somebody is asking, have you observed any limitations in the size of the oligo that can be analysed? Do you have any more information?
So I can say that we have analyzed oligos as large as 100 mers, and we have observed peaks, and we have successfully deconvoluted that data to get the right mass. We haven't really explored oligos that are bigger than that yet, but I would imagine that the constraint would be more on the mass spec side than the ZipChip side. So as long as you have the right mass spec method that goes with the separation method, I would imagine that you can easily analyze larger oligos as well.
Okay, good. Okay, another question. Somebody asks, can you resolve shortmers well? For instance, 9 mer N1 from FLP 10 mer or 19 mer from 20 mer? What would you say?
Yeah, so that's sort of a repeat of a previous question. And the answer was, it depends. Not necessarily every time. It's easier on the shorter example, but because the separation is not fundamentally length-based, it depends.
Great, thank you. And another question, can you separate deamidated species?
Do you want to take that?
Yeah, I'll take that. I don't know that we've specifically looked at that yet. If it alters the charge, then very likely yes. But I don't think so far in the example work that we've done that.
Okay, thank you. Okay, another question. Did you observe the widened peak induced by phosphoroethate linkage replacing phosphodiester linkage? Hopefully I pronounced that correctly. Any answers for us?
So the data that Adi showed was actually a mix. He had some examples of phosphorothioated species and some that were unmodified completely. And in our conditions, we did not see significant band broadening associated with that.
Okay, thank you. Okay, so some more questions. Oligos are usually analyzed using negative mode ESI. Can you tell us, do you lose sensitivity when switching from positive to negative mode? What more can you tell us?
So I can take that one. The short answer is no. We don't lose sensitivity by going from negative to positive mode. In general, you get more efficient ionization efficiency in positive mode on the mass spec. And as I mentioned in my presentation, the number of charge states observed in negative mode data is higher, and the peak intensity is distributed among those charge states. And as you saw from the data for the 20-mer, we get fewer charge states in positive mode data. We've demonstrated that the LOD for the 20-mer is 5 nanomolar, which makes the method pretty sensitive. And of course, the sensitivity differs as the length of the nucleotides in the nucleic acids changes.
Great, thank you very much. Okay, another question. Somebody asks, are there any plans to use this on Waters or Agilent MS? What more can you tell us?
Not in the immediate future. We are vendor neutral in terms of our compatibility with other mass spectrometers, so there's nothing preventing us from doing it at some point, but there's currently no instruments coming out anytime soon that will do that.
Great, thank you very much. Okay, moving on, another question. Somebody asks, how much oligo material is required for a single injection? Who can take that question?
Do you want to take that, Scott?
Sure. So from just a volumes of samples standpoint, the ZipChips use for actual analysis a really tiny, like nanoliter injection. But from a practical standpoint, from the what goes into the autosampler and what gets transferred into the microfluidic device, 10 microliters is the minimum aliquot that it can sample. So that's really the limit, the sort of 10 micromolar or greater, and then Adi gave the dynamic range information, so sort of low nanomolar up to sort of low mid micromolar concentrations.
Good, thank you. And some more questions. Somebody asks, did you observe adduct ions? What more information can you give us on that?
Yeah, I think I already showed in the data for the 20-mer and some of the other molecules that we do see alkali metal ion adducts, but the dominant charge state is, or the dominant adduct is the protonated adduct.
Excellent, thank you. Another question. Which sample concentration is needed for 20 to 40 mR samples? And do you have LOD values?
Yeah, so that's another one
Yeah.
that I think Adi covered directly. So yeah, for the 20 mR that Adi showed, Adi, you can give the numbers again.
Yeah, I think it was 5 nanomolar for the 20 mR, and we've also done these kind of studies for 40 mR, and we were able to see down to 10 nanomolar for the 40 mR.
Excellent, thank you. Okay, moving on to another question. How long does the system take to switch from an oligo method to protein analysis method? Who can answer that one?
I can take it. For switching between methods, you would just have to remove the chip, and then you prime the autosampler and a new chip with the new background electrolyte solution. And that overall process should take no more than 15 minutes. And you should be able to start analyzing your protein samples right away. Since we don't use any ion pairing agents, there are no extra steps required for cleanup and removal of those ion pairing agents. So the downtime that is associated with use of ion pairing agents is eliminated. So I would say it should take no more than 15 minutes.
Brilliant, thank you. Okay, another question. What kind of data processing software can you use for analyzing ZipChip data?
So the data that is generated on the mass spec vendor platform and is also stored using the existing software from the vendors. There is no special software required for data processing, and that is ZipChip specific, right? So you can use the existing software. ZipChip has its own method acquisition or data acquisition software just for the separation part. But for data processing, you can use the vendor software and also some other third-party softwares, such as Protein Metrics.
And I'll just add that although most methods are negative ESI based, it is not the case. I mean, so the software that's out there, from what we've seen, generally can also handle positive ESI data for this application. So we know we've been working with Biopharma Finder for the Thermo software, and I believe the same is true for Protein Metrics software.
Fantastic, thank you. Okay, we have just one more question to run through, and that is, how many samples can you analyze per chip? What more information can you provide?
So each chip can analyze up to 125 samples. You can certainly analyze a smaller number of samples on one day, and then dry the chip out, and then come back in a few days and reuse that chip. But the maximum number of injection count is 125 per chip.
Excellent, thank you very much. Okay, well, unfortunately, that's all the time we have for today. Please join me in thanking Adi Kulkarni and Scott Mellors for their time today. Thank you so much for answering those questions. Thank you.
All right, thank you all.
So a message for the audience: as you exit the platform, a survey will appear on your screen asking you to rate today's webinar. Please take a moment to provide your feedback, but if now is not a good time, the survey will be sent to you via email very soon, so please complete it when you can. On behalf of European Pharmaceutical Review and 908 Devices, I'd like to thank you all for attending, and we hope to see you at another webinar soon. Thank you and goodbye.